Hello and welcome to Sam Biology class. Friends, today we shall discuss about different types of DNA repair. Before that, I request all of my friends, please subscribe my YouTube channel and watch, like and share my videos with your friends. DNA is the genetic material. Its integrity is under constant onslaught from radiations, chemical mutagens and spontaneously arising changes. In spite of this, the rate of mutation remains remarkably low. This is due to the efficiency with which DNA is repaired. Many complex pathways involved in DNA repair are mismatch repair, direct repair, base excision repair and nucleotide excision repair. Most of them depend on the presence of two strands because nucleotides in the damaged area are removed and replaced. Out of four, mismatch, base excision and nucleotide excision repair involve the replacement of nucleotides, whereas in direct repair, nucleotides are not replaced. Repair mechanisms that involve the nucleotide removal utilize a four-step common pathway. First step is detection. First step is detection. Second step, excision. Third step, polymerization. And last step is ligation. The primary differences in the mechanism of mismatch, base excision and nucleotide excision repair is in the details of detection and excision. All three utilizes DNA polymerase. All three utilizes DNA polymerase and DNA ligase to fill the gap produced by the excision and removal of damaged nucleotides. Last one is mismatch repair. Replication is extremely accurate. Each new copy of DNA has less than one error per billion nucleotides. However, in the process of replication, some mismatched bases are incorporated into the growing DNA chain with a frequency from 10 to power minus 4 to 10 to power minus 5. Some of these replication errors are corrected in proofreading. By DNA polymerase. Many of the incorrectly inserted nucleotides that escape detection by proofreading are corrected by mismatch repair. After the replication error has been recognized, mismatch repair enzymes cut out a section from the newly synthesized DNA strand and fill the gap with new nucleotides by using original DNA strand as the template. Second one is direct repair, in which the altered nucleotides are not replaced. Instead, they are changed back into their original correct structure. And most well-studied example of direct repair is photoreactivation. of UV-induced pyrimidine dimer. Another pathway is base excision repair, in which a modified base is first excised and then the entire nucleotide is replaced. The excision of modified base is catalyzed by a set of enzymes called DNA glycosylases. Each of which recognize and remove a specific type of modified base by cleaving the bond that links a base with the first carbon atom of deoxyribose. Last one is nucleotide excision repair that remove bulky DNA lesions like pyrimidine dimers that distort DNA double helix. Nucleotide excision repair is quite versatile. It can correct many different types of DNA damage. This pathway is found in the cells of all organisms from bacteria to humans. Nucleotide excision repair pathway is complex. In humans, many different genes are involved. First, a complex of enzymes scan the DNA looking for the distortion of its three-dimensional configuration. After the distortion has been recognized, many different enzymes such as helicase, single-strand DNA binding protein, DNA polymerase, and DNA ligase act in a specific sequence to repair the damage. A common type of DNA damage is double-stranded break, in which both DNA strands are broken. Double-stranded breaks are caused by ionizing radiations, oxidative free radicals, and other DNA damaging agents. Double-stranded breaks are particularly detrimental to cell because they stall replication and may lead to chromosome rearrangements, such as deletion, duplication, duplication, inversion and translocation. Inversion and translocation. And translocation. There are two major pathways that can repair double-stranded break. One is homologous recombination. One is homologous recombination. Homologous recombination. And the other is non-homologous and joining. Non-homologous and joining. So friends, this is all about different types of DNA repair. For more updates, subscribe my YouTube channel, Sam Biology Class.